Instrumental insemination is an essential tool for both research and stock improvement. Our increasing knowledge of bee genetics and the advances in molecular techniques offer new opportunities in bee breeding. Improvements in equipment design provide high repeatability and personal choice in techniques. With proper treatment and care, instrumentally inseminated queens perform as well as naturally mated queens. The insemination procedure is presented here in step-by-step detail with troubleshooting sections to help you avoid common mistakes. My goal is to help you utilize instrumental insemination and take advantage of the potential this offers. Numerous drones from diverse genetic backgrounds. Instrumental insemination provides the ability to control mating, enabling the study of bee genetics and the development and maintenance of selected stocks. With the sequencing of the honeybee genome comes exciting new opportunities. We are beginning to identify the function of specific genes and may learn how to turn these genes off and on to control diseases and parasites. To ensure success with instrumental insemination, every step of the procedure, from the rearing of queens and drones to the establishment of laying queens in colonies, is to provide optimal queen and drone rearing conditions. This requires strong, healthy, well-fed colonies that are free of pests and disease and chemical treatments and residues. Drone production is just as important as queen rearing. Drones, which are seasonally produced, have a high rate of attrition and are sensitive to stress. Obtaining a plentiful supply of mature drones is often the limiting factor in an insemination program. To save labor, virgin queens and drones can be banked in nursery colonies, but keep in mind that the queen can be subject to injury from aggressive results. Use quality rearing conditions. Establish each virgin queen in a nucleus colony. Queens can be caged as they are extremely active and will take flight. The first step is to select drones for semen collection. A flight box provides easy access to drones. Drones have a high rate of attrition, and losses can occur during rearing, maturing, and mating. To prepare for semen collection, first make sure the syringe is properly prepared. Draw a small airspace into the tip of the syringe and make sure the plunger is drawn forward to allow sufficient space for semen collection. Collect approximately half a microliter of saline in the taper of the tip. Bring the drone's endophallus and the syringe tip into focus. To position the queen in the holding tube, first place the queen head first into the backup tube. Hold the backup tube flush against the queen tube. The queen should walk into the tapered holding tube. Place this over the carbon dioxide valve and properly position. To open the queen, use both hooks simultaneously. If you have difficulty opening the queen, the ventral hook may be inserted first, followed by the sting hook. The ventral hook can be turned sideways and used to stabilize the sting. When the sting is threaded, move the ventral hook back into position and lift the sting with a perforated hook. Once the tip is properly positioned, inject a small drop of saline along with a small airspace in the taper of the tip. This allows you to test if the valve fold has been bypassed. We see the spermatheca of a virgin queen, which is clear and transparent. This is the spermatheca of a mated queen. Notice the marbled creamy color. If the spermatheca contains only a small amount of sperm, the appearance of the spermatheca is cloudy, as seen here. The optimal condition is to directly release the queens into nucleus colonies. Queens should be introduced as queen cells or virgin queens, and for convenience can be caged. To facilitate acceptance, spray the queen and bees with a fine mist of sugar water. A review of studies from 1946 to the present comparing instrumentally inseminated queens and naturally mated queens clearly demonstrates the competitive performance of instrumentally inseminated queens and the importance of proper care. Efficiency in the technique of instrumental insemination requires practice and attention to detail. The first attempts may seem awkward and frustrating. Concentrate your efforts on mastering each step of the procedure. Success and efficiency will develop with experience. Efficiency in the technique of instrumental insemination requires practice and attention to detail. The first attempts may seem awkward and frustrating. Concentrate your efforts on mastering each step of the procedure. Success and efficiency will develop with experience. To achieve this requires proper insemination techniques, beekeeping skills, and proper pre- and post-insemination care of queens. Instrumentally inseminated queens are capable of heading productive, full-size colonies. Insemination is a valuable and essential tool for honeybee research and stock improvement. With the increasing challenge of pests and disease, selective breeding offers the best long-term solution. Sophistication in instrument designs makes the technique easier to learn and highly repeatable. Add to this our increasing knowledge of bee genetics, breeding system designs, and selection methods. Instrumental insemination truly offers an exciting future potential.